Good afternoon. It's a pleasure to be here today. My name is Angela Columbano. I'm Head of Business Development and Partnership at Genethon. It's a pleasure to spend the next 15 minutes talking about exchanging with you about Genethon. This is a non-for-profit, let me just manage to, It's a non-for-profit organization that was created by a patient association. It has been a pioneer in gene therapy for the last 30 years. We are among the originator of Zolgensma, commercialized today by Novartis. We have been a pioneer in the bioproduction of viral vectors since the late 90s. And we have been very successful in building partnership with both private and public organizations. And all of that is based on a very, very sound science with over 1,500 articles published in peer-reviewed journals. So we have an organization that, although it's non-for-profit, is really structured as a fully integrated biotech company with capabilities from bench to bedside. We have a team of about 100 scientists, where the Genethon scientists work hand in hand with academics to really catalyze breakthrough innovation. We also have an in-house, quite sizable CMC development team, and we're going to talk a little bit about that later in the presentation. And we have also a fully integrated clinical, regulatory, medical team that is today in charge of our clinical programs. So we have today a focus mainly on AAV-based gene therapy. We have a pipeline of products in development for the muscle. And the most important, more advanced program we have in-house today is on Duchenne muscular dystrophies. And I'm going to spend a little bit of time describing the data we have generated so far and where we are with this program, what are the next steps. So I think you all know Duchenne. This is a devastating disease that affects boys. On the left-hand side, you see a quite iconic video for our organization. These are the children of the founder of the patient association. And you can recognize the key feature of the disease around 4 to 6 years old. When the disease is diagnosed, you have these difficulties in walking and difficulties in climbing stairs. So generally, a child with Duchenne is never able to climb the stairs with one step after the other. And on the right-hand side, you see a boy that's been treated with GNT0004. This video was taken 18 months after the treatment. You can see that he has absolutely no difficulties in running up and down the stairs. Today he's been treated, the treatment happened 2 years ago today, and this boy has no sign of the disease anymore. So GNT0004 is an AAV8-based microdystrophin gene therapy. We believe that we have managed to assemble together the best possible combination of the microdystrophin that is fully optimized, a muscle-specific promoter, and AAV8. So where we are today, we are conducting a phase 1, 2, 3 clinical trials. We have completed the first phase for the selection of the dose. We had tested 2 doses, 10 to the 13 and 3, 10 to the 13, and selected the second dose to go into pivotal trial. This is the lowest dose among all the programs that we have today in Duchenne. We have, in terms of the result from the first phase, a very strong clinical result measured with the North Star assessment score. We also have seen significant drops in the key biomarkers such as CK and a very strong expression in microdystrophin. So we just, the most important, certainly, clinical results, which is going to be the primary endpoint of the pivotal trial, is the changes that we have observed in the NSAA. which is the clinical score to measure the improvement in the walking and in raising from the floor and all the parameters related to the motility of these patients. So you can see in the 3 patients that have been treated at the highest dose, 10 to the 13, there is a stabilization and then an increase in the score versus a control arm. And just the difference is 4.7, and it's been shown that a clinically meaningful change in NSA is equal to 2.5, so we're well above. So where we are today, we have completed, as I said, the dose escalation trial. We have selected the dose. We have seen up to 85% expression of microdystrophin, a clear inflection in the NSA score, and a very strong decrease up to 90% of the creatine kinase biomarker. So this is obviously a very, very encouraging efficacy data, and we're going to start the pivotal trial in the next few weeks. So we also have a quite important portfolio of program in liver and metabolism, one of which Krieger and Najjar is today in pivotal phase in Europe. So in the second part of the presentation, I wanted to give you some highlights on our technological platform, because we don't work just on gene therapy product, but also on platforms that can enhance the efficacy and the safety of the next generation gene therapies. So we have proprietary tissue-specific capsids, and I'm going to talk about in particular two approaches we have in-house. We also have a large team that look at the immunoresponse and try to overcome with a different type of strategy the immunoresponse to AAV. And also we have quite a significant investment on enhancing bioprocess. So we have different approaches with regards to improving the tropism and the efficacy and then for the safety of capsid, rational shuffling, and then a computational rational design that is combined with a library generated with artificial intelligence. With regard to the rational shuffling, this is based on selecting backbones with a low seroprevalence and the insertion of certain regions that can drive the organ tropism. Based on this rational shuffling, we have created a large library of capsids that have a specific high organ tropism, low seroprevalence, and high manufacturing yields. So this is just an example of one of the capsids that has been generated through this approach. Is a capsid that has a very interesting tropism through the CNS. You can see on the right-hand side that you have, when compared to AAV9, a similar transgene expression in the brain, but is significantly lower in the liver and in the heart, and also in the dorsal root ganglion, DRG, that is important in terms of safety. And this data have been produced in mice and then confirmed in non-human primates. So this is the first platform. The second platform is based on computational rational design. So here we start from identifying a specific receptor in the tissue that we would like to target, and then through artificial intelligence predictive methodology, we find the right sequence of the variable region of our capsid to bind the specific receptor in order to have high tropism, high stability, and so high yields, and low seroprevalence. And the result of this approach have been published a few months ago on the Nature Communication. And the first capsid that has been generated through this platform is a capsid that targets the muscle. And here you can see on the left-hand side the interesting biodistribution where LICA, this is the name of the capsid, has a significant high tropism in the muscle versus the liver. It's 660 times more, better biodistributes in the quadriceps versus the liver. We also tested in disease models, and at low dose we have a very high transduction in the muscle. And finally, we also have interesting data in non-human primate, where you can see that we have improved versus AAV8 the transduction in the muscle up to 34 times, and a significant strong decrease in the liver. So again, the NHP data confirm what we had seen in the rodents. So I also wanted to emphasize how since probably the last 30 years, Genethon has been among the first organization working in gene therapy that has prioritized the importance of reducing cost, increasing yield, and increasing quality in our bioprocess. So we have an in-house team of 40 experts. We have developed up to 14 different bioprocess for 14 different programs, and we can go up to a large scale in-house. And the whole GMP manufacturing of Genethon was spun off some years ago to what has become today Ipos-Casey, that is an SK Pharmateco company, and is one of the largest CDMO platform for gene therapy in Europe. So in the last few minutes, I just wanted to give you a quick summary of the last project that Genethon has launched together with a large number of partners. This is a gene therapy biocluster that has been encouraged and financed by the French government. It's called Genethon. It was created last year. We are just in the first steps of launching this very ambitious organization. Today, 7 founders have created Genethon. Genethon was really the catalyzer, together with other private and public organizations like INSERM and Spark Therapeutics and Ipos-Casey. We have today about 30 partners and a large number of stakeholders, and we are really looking to increase and enhance this ecosystem not only through French, but within international players. So the mission of this biocluster is really to accelerate innovation and gene therapy development. And when I say gene therapy, is the broadest possible definition of gene therapy. Is therapy based on DNA, on RNA, and on gene editing, and all enabling technologies. So we have 5 key missions. The first is really to put together within the same ecosystem French and international leaders for gene therapy, is to finance a certain number of technological platforms that are open to the ecosystem and that can facilitate the development of gene therapy, is to accelerate partnerships, is to create a training module that is dedicated to all the expertise and jobs around the gene therapy, and finally an incubation program for promising project and young startup in gene therapy. So I just wanted to give you a flavor of the platform that we are going to finance in the next coming months, and these platforms are really located in institutions for non-for-profit or academic, with the idea they are really open to any player that is willing to enter into the gene therapy or is looking for innovation and tools to build the gene therapy programs. So there's a number of preclinical platforms dedicated, for example, to gene editing QC lab, to the design and the testing of novel AAV capsid, to the design and testing of novel non-viral capsids, so for example LNPs, as well as to predictive novel preclinical models. We also have some bioproduction platform, a quality control for AAV, and an ex vivo GMP manufacturing, and a number of platforms that should help the clinical development. For example, structuring of large patient database, also improving biomarkers validation, as well as other type of tools that can help design and conducting clinical trial. So we are open. We are open for business. We are looking for any type of institution, private or public, that is interested in gene therapy to join us, either by following us or by entering into the association, or by participating to the creation of the access to platforms. Thank you very much.